Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and we're going to continue our lesson today on genetics. Specifically today, we're getting into Punnett squares, which y'all might remember. These can be used to predict the probability of offspring for a specific crossing of parents. Okay, I'm going to show you how to set it up, what it can be used for, and we're even going to practice a couple towards the end of the lesson. So let me get y'all's attention up on the board. Thank y'all so much for making it here today, and we'll go ahead and hop into it. So what is a Punnett square? Well, a Punnett square is a tool which can be used to predict the outcome of a cross by displaying all possible combinations of gametes. Okay, this is an example of a Punnett square on the board here. Okay, a couple of things that you may want to know. We usually put the father on the top and the mother on the side, although that's usually not going to determine if you get the question right or not. What I do want you to know is that on the outside of the box, you can see the capital A and the lowercase a on the top in the pink up there. Those represent the gametes, okay? So on the top and the side, those are the gametes, and then inside of the square, inside of the Punnett square, those represent those gametes coming together, and so every square in a Punnett square represents an individual, okay? On the top and on the side, those represent gametes, okay? So Punnett squares. Punnett squares show the expected proportion of possible genotype in the offspring of a cross, considering random fertilization. It displays all possible combinations of gametes and the likelihood of each combination, and it can be used to determine the probability of a specific genotype or phenotype occurring. A lot of times when you do these Punnett square problems, you're going to be asked to give the genotypic ratio and the phenotypic ratio, and so I just kind of wanted to briefly touch on what those are. Remember that genotype talks about your allele combination. So what are the letters that you have? Do you have two capital letters, two lowercase letter, one of each? That would all be considered your genotype. Your phenotype, on the other hand, is the physical expression of those genes. So are you tall? Do you have brown hair? Are you left-handed? Those would all be considered a phenotype. And they are not often the same, okay? Because remember, with genotype, there's a difference in between that homozygous dominant and the heterozygous. But in the phenotype, we wouldn't be worried about that difference, because a heterozygous individual and a homozygous dominant individual is going to have the same phenotype because they have at least one of those dominant alleles. So it can be a little tricky when you're having to differentiate in between these two terms, okay? So here's the first step in making a Punnett square. Step one, you want to determine the gametes from each parent, okay? And so this is kind of like a breakdown of it, okay? On the left, we have the possible genotypes, and then on the right, we have what gametes we might be getting from them. So at the top, if your genotype is big B, we would say that that is homozygous dominant. Well, your possible gametes, you can give either one of the capital B's, okay? Because they are being sorted independently. Now where it gets a little tricky is this one in the middle, okay? Big B, little b. We would say that this is heterozygous. And the possible gametes for this individual would be either one of them. We could give the big B or the little b, but we can't give both, okay? And then at the bottom, little b, we would say that this is homozygous recessive, and their possible gametes are either one of the lowercase letters, but remember that you can't give both, okay? The reason I'm really harping on that is that remembering that law of segregation is going to be a really big deal towards the end of the unit when we get to pedigrees. So just kind of remember that for me. All right, so here's step two. You want to draw a grid or a box with 4 squares inside of it, okay? Then you put your genotype or the allele combination of one parent across the top and the allele combination of the other parent down the side. So again, usually we would put the father up top. These represents the different gametes, okay? So the big B would be one sperm, the little b would be a different sperm, okay? And then on the left, we have the mother's genotype, but these both represent different gametes as well. So this would be one egg, this would be a different egg. Depending on what egg or which sperm is used to fertilize and make the zygote, that's going to dictate or determine what traits you get. Okay, so these represent different gametes. These are 2 gametes, these are 2 gametes. Okay, Then you just fill in, you pull them down and you pull them across. So what I did was I took that big B and I put it in the 2 boxes below it. And I did the same thing with the little b over here. I took that little b and I brought it down into those 2 boxes. Then I came over here to the side and did the same thing. I pulled this little b and pulled it over and put it into each of the boxes, and same thing with that. And then this is what I'm left with, okay? So you want to pull those allele combinations down and across into the corresponding boxes, and you always want to write the capital letters first. Okay, so if you get a combination like this, it would not be correct to write it as little b, big B. We always want to put the dominant letter first, and that's going to make our job a lot easier as we keep going with this. Okay, here's the next step. So step 4, you want to use the results of the Punnett square to predict the phenotypic and genotypic ratio of offspring. And just a caveat or a little disclaimer I want to give here. Remember that these are predictions and cannot tell the future with 100% accuracy. And an example of this is, can you call a coin toss 10 times in a row? So a lot of times people see this Punnett square and they see that there are 4 boxes. And so students right away think, well, these represent the 4 children that offspring are going to have. But remember that parents don't always have 4 offspring. So these aren't really all the same, okay? Basically what's going on here is with this specific cross, there's a 50-50 chance that you'll get one of these 2 genotypes. You might get the heterozygous, you might get the homozygous recessive. Because 2 boxes have the big B, little b, and 2 boxes have the little b, it's a 50-50 shot, okay? An example of this is just think about that. This is a 50-50 shot, right? But flipping a coin is 50-50 too. And you can't call a head flip tossing a coin 10 times in a row. Just because you get 3 heads in a row doesn't mean the next one's going to be tails. And the same thing's going on here. When you have your first child, or when these parents have their first child, a lot of times students will think, well, this is the first box, so this is going to be the genotype of that parent. That's not true. These represent the possible genotypes of that offspring, okay? And just even if you do have an offspring that shows that dominant allele, the next time you have a child, it's also a 50-50 shot, okay? It'll make more sense when we're doing some examples up here. Okay, so let's talk about the genotypic ratio versus the phenotypic ratio. Okay, so remember that genotypic is talking about the actual alleles. So when I ask for the genotypic ratio, you need to show me the letters. So we have 2 that are big B, little b, and we have 2 that are little b, little b. Depending on your teacher, you might want to, reduce this to 1-to-1 ratio, but for now, just for this, it's okay to write it like that as the ratio. Okay, and then the next one, phenotypic ratio. Remember, the phenotypic ratio is asking for the physical trait. And so here we have 2 that are going to show the dominant trait and 2 that are going to show the recessive trait. Okay, great work on that. And then lastly, this has to do with Punnett squares as well. Let's say you don't know what the genotype of an individual is. You can do something called a test cross, and this can help you determine an unknown genotype. So a test cross is used to determine the genotype of a dominant organism by crossing it with a homozygous recessive organism. So if someone is showing the dominant trait, they could be homozygous dominant, but they could also be heterozygous. And you can determine what their genotype is by crossing it with a known homozygous recessive organism. And if you ever see that recessive trait pop up, you would know that this dominant organism here has to be heterozygous. Because if any of the offspring is heterozygous, that means that they're getting one of the recessive alleles from this organism and they're getting the other one from this organism. So a test cross is like a way to determine an unknown genotype, okay? So y'all, I have some practice questions I want to do. I think it'll make a little bit more sense when we start putting pencil to paper. So teacher, if you can, go ahead, pass that sheet out. Why don't y'all take 5 or 10 minutes to work on it, see what you can come up with, and then when you're ready, come back to me, unpause the video, and I'll do it on the board up here for everybody. So take a couple minutes to work on that, and I'll knock it out on the board up here in just a second. All right, hopefully this didn't give y'all any trouble, but just in case somebody's a little confused, I'm gonna do it on the board up here with y'all. Y'all feel free to change or add anything you don't have, and hopefully this will clear up any confusion that we've had. So let's go ahead and jump into this and see what we have here. Number 1 says, in pea plants, tall plants are dominant to short plants. Which of the following represents the genotype of a pea plant that is heterozygous? So there's 2 terms we have to know here in order to get this question right. First of all, we need to know what genotype is. Is genotype the allele combination or the physical appearance of an organism? Well, genotype and genes kind of sound alike, right? And so we're not going to use these terms. We're not going to use tall or short because those would be considered the phenotype. And the question here is asking for genotype. So that's telling me it has to be one of these letter combinations on the right. Okay, next it says a pea plant that is heterozygous. Okay, well this root word here, hetero, does hetero mean different or the same? By now hopefully we know that hetero means different, and so what we're looking for is an allele combination that is different. 2 different letters. And so to me, I think we want to go with letter D here. Big B, little t, that's telling us that this is the genotype of a heterozygous individual. Okay, let's keep it going. Number 2. 2 says, in pea plants, tall plants are dominant to short plants. Which of the following represents the genotype of a pea plant that is homozygous dominant? Okay, so again, this says genotype, so we know we're not going to use these words tall and short because those would be considered phenotypes. Okay, the allele combination is what the genotype is. And then this one says homo, so is homo different or the same? Remember that homo is a prefix that means the same, and so these are going to be 2 letters that are the same to each other, so this should be letter A. Okay, so you can see how knowing these vocabulary terms is going to play a big role when you're trying to answering these questions. All right, let's keep it going, come down here to 3 and go ahead and start practicing on some of these Punnett squares. So 3 says, what combination should go in the missing box? Okay, and so it's this box right here that we're trying to figure out. Well, what we want to do is we just pull the letters down and across into the corresponding box. So you can see that we already have one of the capital letter A's here, but we need to bring it all the way down into this box, okay? And then we're going to do the same thing with this little a. We can see we have one of the little a's in this box, but we need to pull it over into this box as well. So we've got a capital A and a lowercase a. This is what should be in that box right there. And so that means that letter C is going to be our correct answer. Okay, hopefully that's making sense how to fill out these Punnett squares. Okay, let's keep it going. Here's 4. Okay, so we're going to do our first cross now, and it's going to ask for the genotypic and the phenotypic ratio. Okay, so this says cross a plant, cross these 2 genotypes together. We have 2 heterozygous individuals. It says what are the genotypic and the phenotypic ratios. So what we want to do here is we are going to use these 2 genotypes and set up our Punnett square. We're going to set it up by putting one allele over and to the side of each box. So here's the first individual is capital A, little a, so I'm going to write it like that. That's probably the father, and then this might be the mother over here, which is also another capital A and a lowercase a. And then we just fill in our box. Okay, so this one comes down into both of these, this one comes down into both of those, this one comes across into both of those, and this one comes across into both of those. So let's kind of fill it in and see what we can kind of come up with for those, okay? So this is what it should look like. Okay, so once the genotypic ratio first, how many different combinations of genes do we see here? Well, I see 1 that's homozygous dominant, I see 2 that are heterozygous, and I see 1 that's homozygous recessive. So we would write this as 1 to 2 to 1. Okay, that's the genotypic ratio. And we might want to go ahead and include what the combinations are, so we'll write it something like this. And that lets them know that we've got 1 homozygous dominant, 2 heterozygous and 1 homozygous recessive. All right, so now we want to know the phenotypic ratio, and this is going to be a little bit different because we need to ask ourselves which of these 4 boxes would be showing the dominant trait and which of these 4 boxes, would be showing the recessive trait. Okay, well, all 3 of these, let me get my eraser out so you can kind of see what I'm talking about. Okay, so all 3 of these would show the dominant trait. They don't have the same letter combination, but because these 2 are heterozygous, they have at least 1 capital A, they're all going to show the dominant trait here. So we have 3 showing the dominant trait and only 1 showing the recessive trait. So this combination would be 3 to 1. And we can go ahead and write it like, we would write dominant to recessive. So 3 are showing the dominant, 1 showing the recessive. Okay, great, let's keep it going. All right, I got a couple more here. So number 5 says, in mice, black is dominant over brown. 2 black mice mate and have some black and some brown mice. What is the genotype of the parents? Okay, so this one's a little tricky because it's not actually giving us the genotypes of the parents. What it's telling us is that it tells us that 2 are black, and in the offspring, some are black and some are brown. And then it's wanting us to come up with the genotypes of the parents. So here's how I would tackle this problem. I'm going to go ahead and set up my Punnett square like I normally would, and I'm going to use the information that is available to me. I'm going to draw this a little bit bigger so you can see what I'm doing. Okay, big Punnett square here. Okay, so here's what we do know. We know that black is dominant over brown, and it's telling us that 2 black mice mate. Okay, so we don't really know the genotype of the parents, right? We do know what one of the alleles have to be. Because black is dominant over brown, that means they have to have at least 1 dominant allele. So all we can really fill out at this point is this. I'm going to use those 2 capital B's to represent this is, at the top is a black mouse and at the side is a black mouse. We don't know what these alleles are at this point, but we can figure it out by solving the rest of this problem. So let's see what we got here, okay? So I'm going to fill in what I have. All I can really do so far is using these capital B's. So I'm going to fill in what I have, okay? And then I don't really know what's going to go here and what the 2 in these 2 boxes are and what that one is, right? Well, let's see what the question says, and it's going to give us some hints here, okay? So it says, in the question, it says some of the offspring are black and some of the offspring are brown. So let's look at this box, okay? What would I have to put, what combination of alleles would I have to have in this box to have a brown mouse? Well, to show the recessive trait, you have to have 2 recessive alleles. So I know that this box has to be little b, okay? Well, that kind of helps me figure out what the genotypes of the parents are, because that means one of these little b's came from this one, and one of the little b's came from this one, right? So let's use that information and fill out the rest of the Punnett square and see if everything is still true. Okay, so I'm thinking that they're both heterozygous like this. Let me fill in this square. Okay, so let's make sure that everything is correct. Do I have, let's make sure, let's see, do I have 2 black mice? Okay, this individual would be black and this individual would be black. And then it says some of the offspring are brown. Do I have some of the offspring being brown? This one is brown. So this combination, what we figured out here is all correct. So the answer we want is what is the genotype of the parents? So we know that the genotype has to be, for this one, it's got to be letter C. That's the only way we could have 2 black mice that are producing brown parents. So sometimes you got to take that, kind of this roundabout way to figure out the problem. Okay, let's keep it going. All right, so this is number 6. This says purple is dominant over white in pea plants. A heterozygous purple flower is crossed with a white flower. What are the genotypes of the offspring? What are the genotypes and phenotypes of the offspring? Okay, so this says purple is dominant, heterozygous purple. So what would their genotype be? Well, if they're heterozygous, they would be like this, big P, little p. and it says crossed with a white flower. Because purple is dominant, we know what a white flower's genotype is. They have to have 2 lowercase p's, okay? So now I know what the genotypes of the parents are. Let's do my cross and see what I came up with. One little hint is that if you have letters where the uppercase and lowercase kind of look similar, really over-exaggerate the difference in between those. So I know this one is a capital P, everything else is recessive or lowercase, so I'm going to write those recessive ones really small so I don't confuse myself, kind of like this here, okay? And then these are 2 little p's, and these are 2 little p's. Okay, so it wants to know what the genotypes and phenotypes are. Okay, well I see 2 genotypes. We have 2 that are heterozygous and 2 that are homozygous recessive. So let's see if that's in my answers anywhere. I've got 50 big P, big P, big P, little p, and 50% little p, little p. So this is going to be our answer. These are the genotypes. And these are the phenotypes, the purple and the white. So letter B is going to be our correct answer. Okay? Y'all, thanks for hanging in there with me. I know this stuff is confusing. We got a couple more to go though, okay? So let's try 7. 7 says, in barn owls, brown feathers are dominant over gray feathers. Brown feathers are dominant over gray feathers. If 2 heterozygous brown feathered owls are mated, what percentage of their offspring would be expected to have brown feathers? So this one's nice and easy. It's telling us what the genotypes are. It says heterozygous brown feathered owls. So we know that for these, they're both going to be heterozygous, so we can use big B, little b for both parents. So kind of like this. These are the genotypes of the parents. I'll draw my Punnett square and we'll fill it in. Okay, and it wants to know what percentage of their offspring would be expected to have brown feathers. Well, all 3 of these boxes are showing brown feathers, okay? Even those heterozygous, the big B, little b, they're still showing brown feathers because brown is dominant here. So 3/4 is 75%. So letter C is going to be our answer there, okay? Great work, great work. Let's keep it going. I got just a couple more for y'all. Okay, number 8 says cross 2 heterozygous tall plants, tall pea plants together. What is the genotypic ratio? Okay, so cross 2 heterozygous tall pea plants together. Okay, so we know that they're both heterozygous, so they'll be something like this: big T, little t, big T, little t, and then I'll fill in my Punnett square. All right, and it says, what is the genotypic ratio? Well, I've got 1 to 2 to 1. All right, I see 3 different genotypes there. So let's see, for this one, what is the genotypic ratio? All right, this should be 1 to 2 to 1, okay? Great work on that one. All right. So number 9 says, in pea plants, yellow seeds, which is a capital A, are dominant over green seeds, which is a little a. Which of the following would result in yellow seeds? Okay, so it's telling us here that yellow seeds are the dominant ones, and it wants to know which of these 3 would have yellow seeds. Well, we know that this one's definitely going to have yellow seeds because it's got the homozygous dominant, but this one also is going to have yellow seeds. Because it has 1 dominant allele, the yellow seeds are going to show there. Okay, so 2 for that one. Okay, great. So here's our last one, number 10. It says, which of the following is homozygous dominant? So let's remember that homo means the same, and dominant, we're always going to use a capital letter. So which one of these is showing 2 capital letters? That's going to be number C. Okay, so great job there, y'all. Thanks so much for following along. I know this stuff can be tricky, but we're going to do plenty of practice as the unit continues. So thanks so much for hanging in there with me. Your teacher has some other work for you to do, so make sure to give that your best effort, and I hope everybody has a great day.